Welcome everybody. This is our, second to last colloquium of this academic year. And my delight to introduce Earl Miller of MIT. I've been trying to get Earl here for I don't know how many years, but it's always looked up years in advance. So I finally got to say, yes, he's here. Don't let him go. He went to Kent State as an undergrad, majoring in psychology. Then was a grad student with Charlie Gross at Princeton, working on object cells. Then was with Bob Desimone at MIT, studying attention. And has been there for the past 10,000 years.
10,000 years?
How many years?
At MIT? 19 years.
19 years. So, without further ado,
Thanks. Thank you. Well, Peter, thank you for the persistence and thank you for the invitation. It's a really great pleasure to be here. I thought I should begin by telling you a little bit about what we do. So what we do is we experiment in trained macaque monkeys, and we use multiple electrode recording, arrays of electrodes to record in multiple brain areas simultaneously. Here's an example of one of our monkey brains here, an MRI with one of our electrode arrays implanted over the prefrontal cortex. And we often look at electrode arrays within one brain area, so we look at local network interactions within our brain area, and then implant multiple arrays in different brain areas, so we look at macro interactions across different brain areas. And what we do is we train our monkeys to perform a wide variety of cognitive demanding tasks, so we can look at the network properties underlying cognition. And we investigate this on a variety of different levels, from understanding the properties of single neurons. But today what I'm going to primarily talk about is one of the great advantages of using multiple electrodes is you can look at the precise timing of neurons. More specifically, you can look at brain rhythms and how activity in different brain areas and within a brain area synchronize their oscillations, and that gives us insight into the network properties underlying cognition. So I'll begin by asking a very basic question in neuroscience. How do we form a thought or an ensemble? I mean, it's a truism, tenet of modern neuroscience that an ensemble is a subset of neurons that represents a given perception, thought, memory. It has to be that way because a specific thought is going to be some subset of neurons in your brain, right? So that's what an ensemble is. But the problem isn't so simple, especially at the higher levels of cortex. When you get to areas like the prefrontal cortex, which is our main area of study, we recorded multiple brain areas, but we mainly focus on the prefrontal cortex because of our interest in high-level cognition. But we know from many years of studying the prefrontal cortex that neurons there don't just seem to participate in just one ensemble. Many neurons in the prefrontal cortex, in fact, the majority of neurons in the prefrontal cortex seem to be multifunctional. They have what we call mixed selectivity. And that is, you record from an individual neuron in the prefrontal cortex, and you'll find that that neuron will do different things in different tasks, or even do different things at different times in the same task. Like a given neuron could be a signal what color the animal is looking at one point in the task. Then later on in the trial, the neuron looks like it's a motor neuron signaling right versus left. Now, this work came primarily from when we study the prefrontal cortex, we record randomly from neurons using our electrode arrays. And we've discovered over a number of years, we found that when we do so, we activate like 30% of randomly selected neurons in the prefrontal cortex. When we train a monkey on a given task, and 30% of the neurons in the prefrontal cortex have this property that we're looking for that we trained our monkey to do. So there's two possibilities here. Either you fill up the monkey's brain when you teach it three things, because you're co-opting 1/3 of the brain by one thing, or what we suggest is that you have a lot of multifunction neurons in the prefrontal cortex. And we did subsequent experiments to show exactly that by training monkeys to do a variety of different things. And you see the same neuron acting like a different neuron in different tasks. Now, last year, Stefano Fusi's computational lab showed that you need to have these mixed, what he called mixed selectivity neurons, these multifunction neurons, to build a complex brain. Without multifunction neurons, you have a brain that only can learn a very limited amount of things and only can learn very simple things. So these mixed selectivity neurons seem to be give the brain extra horsepower for computation related to cognition. It provides a multidimensional, multivariate space where a lot of high-level processing can go on. But this introduces a bit of a problem. If this is true, what it means is that neurons, especially in a higher level cortex like the prefrontal cortex, don't participate in one ensemble or another ensemble. They have ensembles lying on top of ensembles, lying on top of ensembles. Many neurons participate in multiple ensembles. So if that's the case, how do you activate one ensemble for one thought? I mean, if it was just anatomy, if anatomy was the only thing in the brain that determined which neurons fire, well, then if you try to activate one ensemble, the activity will run to all the other ensembles that its neurons also participate in, and you have a jumble of thoughts trying to be activated. So how do you activate one thought, given that many ensembles share neurons in common? Well, one possibility, not our idea, but idea that originally from Wolf-Singer, Andreas Engel, Pascal Fries, they've suggested that the way way ensembles form is you essentially bind members of the ensemble through synchronized oscillations. We all know that the brain has lots of oscillatory activity. That's what these brain waves are. So the idea is that if you have neurons that participate in two different ensembles, then the way you activate one ensemble or the other, the way you have one ensemble forming one thought, is the neurons that are in the same ensemble synchronize their oscillations, and neurons that are participating in a different ensemble synchronize their oscillations. So by virtue of the fact you have different patterns of synchrony, you can have two different ensembles, even though they share many of the same anatomical connections. So that's the idea. We sought to test this by training monkeys on a task which they had to either pay attention to the color or orientation of a color-oriented bar. So the way the experiment went is the monkey was trained to fixate a spot of light. Then the first thing that happened is we set up the task by having a target dot appear on the left or the right. Now the monkey wasn't allowed to move its eye yet. The monkey was trained to fixate the spot of light here, and that's what the circle represents, the monkey's eye position. It didn't actually appear on the screen. During this preparation period, we also cued the monkey which rule it should follow, whether it should pay attention to color or pay attention to orientation. Shortly after that, after a variable interval lasting from 86 to about 500 milliseconds, the stimulus actually appeared. And the stimulus could be a vertical bar or a horizontal bar, and it could be red or it could be blue. The monkey's job was to move its eyes in one direction for one color. In this case, move to the right if the stimulus is blue. Move to the left if the stimulus is red. If the stimulus was vertical, the monkey had to look left. If it was horizontal, the monkey had to look right. Now, we set up the experiment so most of the trials, 70% of the trials, the two different modalities gave the opposite behavioral response. So we know the monkey really had to focus on either color or orientation and ignore the other modality, or it would make a mistake. So the important thing here is first the monkey gets a rule preparation period where it's cued to which modality is important, color or orientation. Then the monkey actually sees a stimulus and executes the rule. That's the basic design of the task. Now again, what we're looking for here, we're going to look across pairs of electrodes. In this case, we're going to be recording from the lateral prefrontal cortex, right about here, in the monkey's brain. And we're looking for whether different pairs of electrodes show different patterns of synchrony for these two different rule ensembles, color versus orientation. So we're looking for this. And here's what we found. Let me walk you through the slide, then I'll show you what the results are in cartoon form, just to make sure. This is a plot of this. Now we're looking at synchrony between local field potentials across pairs of electrodes. And what's shown here on the y-axis is frequency. On the x-axis is time during the trial. So here's the rule preparation period where the monkey got the rule cue. Then right here where the red vertical line is when the monkey saw the test stimulus, had to execute the rule. Then where the green line is when the monkey made its motor response, which is a saccade. What we found is about half of the randomly selected electrode pairs in the prefrontal cortex showed rule-specific synchrony. They showed greater synchrony for one rule, color, orientation, and while other pairs of electrodes showed greater synchrony for the other rule. About half the electrode pairs showed greater synchrony for the orientation rule, and the other half of the electrode pairs showed greater synchrony for the color rule. And that's what I plotted here. Here on this half are some data from electrode pairs that showed greater synchrony for the orientation rule, and here's the other half of the electrode pairs showing greater synchrony for the color rule. Pink indicates stronger synchrony for color, blue indicates stronger synchrony for orientation. And what we saw, we looked across all frequencies, and what we found is that the main action was in the beta band. In this range here from about 15 or so hertz to about 40 hertz, and maybe even a little bit above, many electrode pairs showed a difference in this frequency of synchrony for one rule or the other. Okay, so this is essentially what happens in cartoon form. We're having the monkey pay attention to color, and here's our array of electrodes in the prefrontal cortex, and we're looking across different electrode pairs. And what we found is there was one set of electrode recording locations that showed greater beta synchrony whenever the monkey was attending to the color of the stimulus. And then on other trials, where the monkey was cued to pay attention to the orientation, we saw different pattern of electrode pairs show increased beta synchrony. So what we found is just what we thought we were hoping to get, which is rhythm-based ensembles, different patterns of beta synchrony for attend to color versus attend to orientation, when the monkey's actually paying attention to color or paying attention to orientation. So it seemed to support this hypothesis that you can actually form ensembles by virtue of rhythmic synchrony. Different members of an ensemble link up when they synchronize together, and you get different sets of neurons synchronizing for one rule, a color, versus the other rule, orientation. And this is what they actually look like across the cortex now. So here's a plot of the monkey's lateral prefrontal cortex. This is the arcuate sulcus. This is the principal sulcus. This is anterior. That's posterior. It'd be about right here on my head. And what the circles show are the recording sites where the electrodes were, where our electrode array was. And the colored lines show connections between pairs of electrodes that showed rule-specific changes in synchrony, showed a significantly greater increase in synchrony for either color or orientation. These are the electrode pairs that showed a greater increase in beta synchrony for color, and here's the electrode pairs that showed a greater increase in beta frequency for orientation. Now, one thing you notice is that these are two unique patterns, so you get unique ensembles for the two different patterns of synchrony, but also there's a lot of overlap, as we predicted. Many of the recording sites, in fact, many of the neurons, this is, so far what I showed you is all local field potentials. If you look at individual neurons, you see individual neurons, the spikes will synchronize to the local field potentials. And there's many neurons, these mixed selectivity neurons in the prefrontal cortex, that will be both color selective and orientation selective. They'll be color selective on one trial, orientation selective on the other trial. And what happens is the neurons that are color selective, they synchronize to the color rule network when the monkey is doing color, and the neurons that are orientation selective synchronize to the orientation network when the monkey is doing orientation. So these patterns actually go all the way down to the level of individual neurons. But true enough, there's some neurons that either only did color or only did orientation. They synchronized to their appropriate network. Many of the neurons did both color and orientation, and they just synchronized to whichever network was currently relevant. So we see evidence for this overlap of ensembles, and also this parsing, these activation of two different ensembles despite this overlap by virtue of rhythmic synchrony. So that's some fun evidence suggesting this. Now, what I've shown you so far is the rule execution period, where the monkey's actually looking at the colored-oriented bar and making his decision about red or vertical or blue or horizontal. Now let me turn to the earlier phase of the task, the rule preparation period, where the monkey's actually getting ready to execute the rule, has been cued to the rule, but hasn't seen the stimulus yet, so can't execute the rule. What happens there? Well, so now we're going to focus on the rule preparation period. And what happens is, some of you people who are fans of oscillations in the brain may have noticed I blanked out the lower frequencies here. That's because we found something in the rule preparation period in the lower frequency range, in the alpha range. And what we found is that only one of the ensembles showed alpha synchrony during the preparation period. The other ensemble did not. So here's our beta-defined orientation ensemble. Here's our beta-defined color ensemble shown here. Only the orientation ensemble showed alpha synchrony during the preparation period. There was no alpha synchrony in the beta-defined color ensembles. We have asymmetry here. Alpha in the orientation ensemble, no alpha in the color ensemble. But it gets even weirder. If you look at the color here, so remember pink is stronger color synchrony, blue is stronger orientation synchrony. The beta-defined orientation ensemble showed alpha in the preparation period, but only did so when the monkey was about to do the other modality, color. Okay? So the orientation ensemble showed alpha synchrony when the monkey was getting ready to not do orientation, but do the other thing, color. So alpha during color preparation in the beta ensemble, but no alpha at all in the color ensemble. Why is that? Why this asymmetry, and why would they show this opposite effect? So again, just to show you make it clear in cartoon form, when the monkey's preparing to attend to color, the first thing that happens during the preparation period is the beta orientation ensemble shows a bunch of alpha synchrony. Then when the monkey actually attends to the color, the beta network is quiet, at least it's non-synchronous, and then the color ensemble activates. But then in other trials where the monkey is preparing to attend to orientation and then attends to orientation, the beta orientation ensemble synchronizes, but there's nothing, no alpha at all in the color ensemble. So why? Why would we see this asymmetry? Well, so that's what this says. Why? Why do we see this asymmetry? Well, the reason is because the monkeys found one modality easier than the other. So what this shows is the monkey's reaction time on the y-axis versus number of trials since switching task on the x-axis. So what the monkey did is the monkey did a trial of one modality over and over again, color, 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 for at least 20 trials, and then we switched the monkey to orientation. And the monkey would do orientation for at least another 20 trials. We randomly varied it so the monkey couldn't predict exactly when the switch was going to happen. But there was these switch moments. So the monkey was doing one thing for a very long time and then switched to the other thing. And what happens is that when the monkey switches from orientation to color, there's an increase in the monkey's reaction time. This is called a switch cost. It's a cognitive stumbling. Sometimes this can happen when the person will make this. You see this in humans and other animals. They will make mistakes sometimes right after a switch. But generally what happens is you get an increase in reaction time. The animal slows down a little bit for this case, one trial, sometimes two trials right after a switch. So we saw this increase. We saw these switch costs, this cognitive stumbling, when the monkey was switching from orientation to color, but we saw no such switch costs, no stumbling, when the monkey was switching from color to orientation. Now what that suggests to us is we have a Stroop-like effect going on here. The monkeys had found it easier to pay attention to orientation than color. I don't think there's anything special about orientation and color. I think it just happened to be the way we drew the stimulus, the stimuli we used. The color wasn't very saturated, and the orientation may have been more obvious. This is an analysis of an experiment we did years ago. I'm not sure, but I predicted if we retrained the monkey, if we we're doing the experiment, we're training the monkeys around, we could switch this by just changing the details of the stimulus. But the stimuli we happened to use, it turned out the monkeys found it much easier to pay attention to orientation than they did the color. So what this suggests to us is that what alpha is doing is alpha is the orientation. Remember, the monkeys found it easier to pay attention to orientation than color. The orientation ensemble is the dominant ensemble here. So on trials in which the monkey is about to pay attention to color, the first thing the brain has to do is use alpha to suppress the dominant network so that the monkey can then use the weaker one. And this is consistent with a wide variety of literature showing that alpha synchrony, increases in alpha power in the brain, is associated with inattention. You disengage your attention from something, you even close your eyes, you get an increase in alpha oscillations. And this suggests that one role for these alpha oscillations may be to suppress networks for potential suppression of certain ensembles. In this case, the ensemble that was dominant to the monkey. So from this experiment, we learned that synchronous beta band oscillations may help form neural ensembles for rules. Alpha may suppress dominant ensembles, and weaker ensembles must be used. So now I'd like to turn to, if there's no questions on that, I'd like to turn to another experiment where we also looked at oscillations, but in a slightly different format. We have a long-standing interest in my laboratory in category learning. We have a number of experiments showing that monkeys can, if If you train monkeys properly, they can learn to categorize all sorts of things, cats and dogs. We train them to recognize small numbers. We train them on concepts like same versus different. So when thinking about how the brain learns categories, there's a couple of observations we were thinking about when we were thinking about how the brain actually learns these things, learns categories. One is that if you look at human fMRI studies, both the prefrontal cortex and the striatum are both activated when humans are doing category learning. And we also noted that there's these anatomical loops between the cortex and the basal ganglia. So the cortex, the prefrontal cortex, for example, feeds information down into the striatum. It goes through an indirect and direct loop in the basal ganglia, back through the thalamus. Then the output of the thalamus targets the same part of the prefrontal cortex that gave rise to the initial projection. That's called a closed anatomical loop. And what closed anatomical loops are good for are recursive processing, bootstrapping. So we thought, well, maybe the prefrontal cortex and striatum work together in order to form categories. It didn't necessarily have to be that way. They could be completely independent learning systems that do their own thing. We thought maybe there's this interactive, iterative process. So we first experiment we did is we recorded from the prefrontal cortex and the striatum, not during category learning, but in a much simpler learning task. And Anita Pasupathy in my lab, she found that there was, if you look at changes in neural activity in the striatum versus the prefrontal cortex during simple associative learning, the striatum changes very, very quickly, and the prefrontal cortex changes much more slowly. The monkey's behavior follows a slower time course of the prefrontal cortex, but the striatum is doing very rapid changes that precede the changes in behavior. So thinking about that, we have a hypothesis that maybe what's going on here is the striatum learns simple things very quickly. Then the output of the striatum, through the thalamus back up to the cortex, the output of the striatum changes these these networks in the prefrontal cortex that have slower changes, slower plasticity. And maybe what happens then is the striatum, by repeatedly setting these patterns up to the prefrontal cortex, what the prefrontal cortex does is figure out what's common among all the patterns fed to it by the striatum. What's the overlap in all the patterns? And what is a category? What is an abstraction? But finding the common elements about a whole bunch of different things. That's what a category is. So the way to think of it is the striatum learns pieces of the puzzle while the prefrontal cortex puts the puzzle together. That's the general idea. And it does so by the striatum slowly training the prefrontal cortex to figure out what the overlap is among all the patterns fed to it. So from that, we made some predictions. If we can record from the prefrontal cortex and striatum during category learning, we predict there should be functional interactions between them. And that isn't a given because, like I said, the basal ganglia is part of this habit learning system. The prefrontal cortex is cognitive. It's cortex. Maybe these two areas are just working independently, trying to solve the task their own way. But if they really are working together in this iterative fashion, we should see some functional evidence of interactions between them. And the other thing this hypothesis predicts is a trade-off. That during category learning, the striatum will begin by memorizing individual exemplars, and then when the animal actually starts picking up on the categories, that's when the prefrontal cortex will take over. When you learn a new category, you tend to start. Imagine you have a little kid when you're learning what a dog or a cat is. Your parent may point to you, that's a dog. And then the first thing you do is every time there's a furry thing, the kid says, oh, dog, dog. And eventually the kid will learn to figure out what's a dog and what's a cat. And what's going on there is the first thing the brain tries to do is just memorize everything. And then eventually it figures out what the commonalities are that make a dog versus make a cat. So what we're suggesting here is that during the initial phase of learning, when everything's just trying to be memorized, the striatum is leading the ball game. And then when the category actually starts to get formed, that's the prefrontal cortex beginning to form the category based on the things the striatum sends to it. So we tested this idea directly by training monkeys to learn 2 new categories each day during 1 3-hour recording session. And what we did is we trained monkeys, for each day of recording, we made 2 categories, category A and B, from prototypes that were generated new on that day. And the prototypes were just patterns of dots on the computer screen. The monkey never saw these prototypes. What the monkey saw was category exemplars made from distorting the prototypes, just by jittering them in space. So the monkey got a bunch of exemplars until finally figured out what the category was. So the monkey learned to categorize exemplars without ever seeing the essence of them in the prototype. They had to figure out the essence by trial and error. And here's the way the task worked: is the monkey began by fixating a spot of light. We showed it one of the exemplars as a cue. there was a memory delay, then the monkey had to make a saccade in one direction for one category, the other direction for the other category. Now, this is not the ideal way to do the experiment. Every other categorization experiment we've done, we train monkeys to do a category match judgment. You have to say yes or no, these two stimuli are from the same category or not. By doing that, you can dissociate the motor response from the category decision, because the motor response means yes or no. It's not unique to either category. We tried to get monkeys to learn a category matching task within a single 3-hour recording session. That's beyond the limits of monkey intelligence. Or it's beyond the limits of the intelligence of the investigators to get the monkeys to do it, but they couldn't do it. So we had to simplify that. We really wanted to get the learning within one 3-hour recording session. So we simplified the task by having this each category associated with a unique saccade to the right, saccade to the left. Not ideal, but it got the monkeys learning within 3 hours. And here's the way we structured the task. And by we, I mean Evan Angelatis in my lab. What he did is he started on the first block of trials. He just showed the monkey 2 novel exemplars, just 2 of the exemplars. And the monkey figured out by trial and error that one exemplar meant look to the right, the other exemplar meant look to the left. Once the monkey figured that out after a few tens or fewer trials, we then added 2 new novel exemplars. So the monkey was performing with the 2 familiar ones, now 2 new ones. And the monkey figured out the 2 new ones by trial and error. And then when we got to the third, when we went to the third block of trials, we now doubled the number of novel exemplars. Now the monkey is getting 4 novel exemplars, and only 2 familiar ones, et cetera, and so forth, until by the end of 8 iterations of learning, the monkey is getting 256 exemplars. Most of them are novel, and he's only seeing them one time on one trial. So the monkey's job, at first the monkeys could just start by memorizing by rote. This stimulus means look right, this stimulus means look left. But as you start adding more and more novel exemplars, now all of a sudden you're overtaxing the monkey's ability to just memorize everything. And the monkey has to go into category mode to continue getting rewarded, continue solving the task, or it's going to fail. And we try to tailor the experiment in such a way so the monkey would succeed on most days. There were a few days where the category prototypes were too difficult and the monkey did fail to learn the categories. But we titrated it so the monkeys would learn on get the categories on most of the sessions. And here's the monkey's behavior. So here's the first block of trials. The monkey's just getting 2 novel exemplars. Chance is 50%. Look to the right, look to the left. Just guess. So the monkey starts at chance, quickly gets up to about 80% correct, which is our criterion. Once the monkey got to 80% correct, we added two novel exemplars. And now the monkey's overall performance is knocked down a bit because the monkey has a chance for the 2 novel exemplars, but it knows the 2 familiar ones, so its performance starts like 75% correct. Then you get to block 3, block 4. Every time you add novel exemplars, the monkey's behavior is knocked down. Until, after a while, the monkey's behavior stops being knocked down. By the time the monkey gets to blocks 4 or 5 here, you're adding novel exemplars, and the monkey's behavior is no longer perturbed. Now remember, at this point, the monkey is seeing more novel exemplars than familiar exemplars. So what that means is what's going on here is first the monkey started out in an SR learning phase where it's memorizing everything. Then, after a while, it starts responding to novel exemplars above chance, and we can say it's now entering a category learning phase where it's starting to get the categories. Therefore, it could deal with a novel exemplar and not just be guessing. Then, after a while, the monkey has now learned the categories. So out here, block 7 or 8, especially block 8, the monkey is at 90% correct from trial 1, even though the monkey's seeing pool of 256 exemplars. Many of them it's only seeing one time on one trial, yet it still knows the category as soon as it sees it. So we can identify these 3 learning phases: SR learning, category learning, and finally category performance that got the category. So the first thing we looked at, if you looked at this is a paper we published a few years ago. We looked at neural activity to the cue. Remember the exemplar of the monkey seeing, and we asked how much information was there about the category or saccade. So one category meant look right, the other category meant look left. And we looked at information during the cue, because if you look at the end of the trial when the monkey's actually making the saccade, that's just a motor signal. What we're looking for is a predictive signal to the cue that tells the system what the correct response is, what the category is, what the saccade is. And this is shown here. This is information on the y-axis. Here's the 3 different learning phases. First, SR learning, then category learning, and finally category performance. And we found the trade-off we predicted. First, when the monkey's in the SR learning phase, the striatum carries much more information about the forthcoming saccade than the prefrontal cortex does. But then, when the monkey switches to category learning phase, category learning phase, and ultimately category performance, now there's a switcheroo. The neural information, the striatum drops down, and now the prefrontal cortex becomes a better predictor of what the response is. So this is exactly the trade-off we predicted. The striatum is leading the game, and you see this is in terms of neural information. You see the same thing if you look at latency for the neural effects to show, is that in the SR learning phase, the striatum leads the prefrontal cortex in latency. Then when the monkey switches into category learning mode, not only does the information go up to the prefrontal cortex, but now they reverse. Now the prefrontal cortex leads the striatum in time. So we saw this trade-off we were expecting for this transition from SR learning to category learning. But now I want to tell you about a brand new experiment we debuted for the first time here, where we analyze this data to look at this question of network interactions, of synchrony and oscillations. So what we found is that as the animal proceeded from SR learning to category performance, there was an increase in synchrony between the prefrontal cortex and striatum, which is what we'd expect, that they really are working together in this iterative fashion. What we found is that when the monkey switched from SR learning to category learning mode, there was an increase in beta band synchrony between the prefrontal cortex and striatum. And we're looking now here, we did the analysis both on LFPs. This is data from LFPs. We also looked at spikes in one area and LFPs in the other, and you see the same, you can see synchrony between the LFPs and spikes as well. But this data is just the LFPs right now. And what this shows is a measure of synchrony between the prefrontal cortex and striatum using phase locking values on the y-axis versus frequency on the x-axis. The black line shows the frequency profile, the synchrony profile, and different frequencies during the SR learning phase. Then the two colored lines shows the category learning in green and category performance in red. And what you see is in this beta band here, there's an increase, a general increase in beta band synchrony between the prefrontal cortex and striatum as the animal switches from category learning to, SR learning to category learning. So increased talking between the prefrontal cortex and striatum once the animal really starts acquiring the categories. The next, so that's what's illustrated here. So we have during SR learning, there's some baseline level of synchrony between the prefrontal cortex and striatum. When the monkey switches to category learning, there's an increase in beta band synchrony between the prefrontal cortex and striatum. Now, what about category performance? There we found something a little bit different. What we found, first in cartoon form, is that at the end of learning, when the monkey finally fully acquired the categories, this general increase in beta synchrony resolved itself to category-specific patterns of beta synchrony. So I showed you in the previous experiment rule-specific patterns of beta within the prefrontal cortex. Now we're seeing the same thing, but between the prefrontal cortex and striatum. Stronger synchrony for some electrode pairs for one category, stronger synchrony for other electrode pairs for the other category. So that's what we found. Here's how we found it. What we did is for each electrode pair, we computed D prime values for the different, this is a measure used by Diane and Abbott, D prime value for the difference in synchrony in the phase locking values between the two categories. So what do we have here? We have three different electrode pairs. This electrode pair shows a stronger synchrony between the prefrontal cortex and striatum for one category. Pair 1 does. Pair 2 shows a stronger increase in synchrony for category B, and pair 3 shows no change at all. So the D prime will be larger in pairs 1 and 2 than 3. Very simple, very straightforward. So, and this is the data. So now we're looking at, here's again, frequency on the x-axis. But now we're looking at our D prime measure of category selective prefrontal cortex striatum synchrony. So we're looking for how much information is there, how much information there is about the category based on the patterns of synchrony alone. How many electrode pairs differentiated between the two categories based on the level of beta band synchrony. And what you see is that the black line again is SR learning, the green line is category learning, and when the monkey, there's no change there, but when the monkey switches into category performance, when the monkey has truly acquired the categories, now there's a significant increase in category selective synchrony between the prefrontal cortex and striatum. So once again, evidence that the brain is forming ensembles, this time between brain areas, that are defined by virtue of their patterns of synchronized oscillations. Now for you aficionados of synchrony, let me point this out that there was no change, no learning-related changes in synchrony within the prefrontal cortex or within the striatum. These changes were specific to between the two areas. It wasn't just a general increase in synchrony. Synchrony didn't change in either area alone. They only changed between the areas. And there was also no change in oscillatory power. So it was truly a change in synchrony, not just a general increase in oscillations in the PFC. Okay, so now we add to this that we have synchronized beta band oscillations help form neural ensembles for rules. Now we're suggesting that they may help form new functional networks between brain areas, essentially larger ensembles during learning, particularly during category learning. And from these two observations, I would argue, and we would argue, and Tim Bushman and I have argued, that maybe this is the neural substrate for cognitive flexibility. I mean, one of the hallmarks of higher level cognition is flexibility in thought. You can change your mind from moment to moment. You can change what you're doing, change your thoughts on the fly. If which neurons formed ensembles, if networks in the brain only formed by virtue of anatomical connections, how does that happen? How do you change how do you have mental flexibility? How do you change your thoughts, your ensembles on the fly if anatomy is destiny in the brain? So what I want to suggest here is that anatomy is just the road, not just, anatomy is the road and highway system in the brain. What these oscillations may do is direct the traffic on the highways. So anatomy just says where information could go. What determines where information actually does go are these patterns of synchrony that can act as a traffic cop that directs traffic in the rest of the brain. Now, one question you may ask me is that, okay, you know, if this is true, if you form neural ensembles by rhythmic synchrony, I showed you two examples of this, and they both were in beta. In fact, for the two different rules, two different categories, all in the same frequency band, beta. So you may ask me, well, if this is true, if you're forming ensembles by virtue of these beta band, 25 to 30 hertz oscillations, how do you hold two thoughts in mind simultaneously? Well, first of all, you all know that we're all very bad at holding multiple thoughts in mind simultaneously. We have a very limited capacity to do so. But we do have some capacity to do so. That being the case, how do I hold two thoughts in mind simultaneously if they're both defined by the same frequency, synchrony in the same frequency band? If I try to activate two ensembles at the same time, why don't they synchronize together and mush the two thoughts together? How does that work? Well, one idea, first from John Listman, is that what the brain does is oscillate the two different ensembles in and out of phase of one another, so-called the phase coding hypothesis. And John's purely theoretical idea was that what happens is the neurons, the spikes that are part of one ensemble versus the other, line up on different parts of the oscillatory cycle, and essentially the brain does a juggling act. You have two thoughts, and the brain's just juggling them in and out of phase of one another, so it keeps them from interfering with one another. That's the idea. Theory's nice, but let's try experiments. So what we did, and by we I mean Marcus Siegel and Melissa Warden in my laboratory, is they trained a monkey to hold two pictures, two objects, we called them, two objects in memory at the same time, and the order in which they are presented. So the monkey began by fixating the spot of light. Then we presented the first object, in this case an avocado. There was a short memory delay. Then we presented the second object, in this case an apple. There was another short memory delay. And then at the end of the trial, the monkey saw three pictures shuffle around in some random order. And the monkey's job was to make eye movements only to the two pictures it saw before earlier in the trial and in the correct order. So in this case, it has to saccade the avocado first and apple second and not look at the lemon because the lemon wasn't seen earlier. Now, for this experiment, we used 4 objects every day, new objects every day, so the monkey really wasn't just memorizing patterns of things. And we fully balanced object identity with order. So sometimes the monkey saw avocado first and apple second. Sometimes apple first, avocado second. Sometimes lemon first, apple second. You get the idea. Everything is nicely counterbalanced by order. And we recorded, in this case, in this experiment, from multiple electrodes in the lateral prefrontal cortex, shown here in the shade. This is a monkey brain here. The monkey's eye would be looking out here this way. So this is the lateral prefrontal cortex along the principal sulcus. The recording sites are back in the posterior part of the lateral prefrontal cortex near the frontal eye fields. And we recorded from electrode arrays with 1 millimeter spacing. So first thing we notice is that when we look at just simple power, oscillatory power and local field potentials, we saw, again, around beta band, either high beta, low gamma, whatever you want to call it, around 25 to 30 hertz or so oscillatory synchrony during the memory delays of the task. So here's frequency again on the y-axis. Here's time during the trial. This is when the monkey saw the first stimulus. There's right here, first object there, first memory delay here, second object there, and finally this is the second memory delay, and the monkey made its response out here. So what happened is every time the monkey initiated the trial by fixating the spot of light, there was a big burst of about 30 hertz oscillations in the prefrontal cortex and 3 hertz oscillations in the prefrontal cortex, which went away for the most part, especially the higher frequency stuff, went away when the stimulus was presented, maybe because this big input DC signal into the brain masked the oscillations for a short period of time. But then when the stimulus went off and the monkey was staring at this blank screen, the oscillations returned. Went away again during the object presentation to return during the memory delay. So the prefrontal cortex does oscillate during the memory delays of this working memory task. What about this phase coding effect? So what Marcus did is every time there was he always looked across pairs of electrodes. Never did this analysis on the same electrode because there's too much chance for crosstalk and artifactual synchrony. Always looked across electrodes at least separated by 1 millimeter, so we were sure we're looking at different signals. And what he did is that every time there was a spike on one electrode, Marcus computed the instantaneous phase of the oscillation on all the neighboring electrodes. And from that, he could essentially divide each oscillatory cycle up into a bunch of tiny windows. So here's one single idealized representation of a cycle. And by knowing when the spike occurred and what phase was going on around it, you can actually take one cycle and divide it up into little tiny windows. Now, what neurophysiologists have done for decades is you take a big time window, like a 1 second during the trial or half a second during the trial, and you compute neural information, like I've showed you examples of earlier, over that one big block of time. What Marcus did is rather than just look over a block of time, he divided each ongoing cycle during the delay into a whole bunch of little tiny windows that repeated 30 times a second, and computed neural information in each one of these little windows. And what's shown here is a polar plot from one representative recording session. This is not information, but level of activity of the neuron. And this is a polar plot where you read it counterclockwise. So 0 to pi is the depolarizing phase of the oscillation. And minus pi back to 0 is the repolarizing phase of the oscillation. So we all know that neurons are more active when they're depolarizing. So sure enough, there was more activity during the polarizing phase of the cycle than there is during the repolarizing phase. So that's just pure activity. But now what Marcus did is then he took these, by summing spikes over a number of trials and a number of cycles during the memory delay, he can actually compute neural information about the two objects the monkey's holding in mind. So what I'm going to show you now is data just from this second memory delay here where the monkey's holding the two objects in mind and their order. And I'm going to show you neural information about the first object and neural information about the second object as a function of LFP phase. This is what we found at around 30, 32 hertz. This is, again, a polar plot. So the depolarizing phase of the oscillation, repolarizing phase of the oscillation here. And the dotted line shows the average information across all phases of the oscillatory cycle. The color line shows the actual information. So what you see is there's a gain of information during the polarizing phase of the oscillation for this first object. It's about a 16 to 25% gain in information over the opposite repolarizing phase of the cycle. But you notice that the center for this information bump for the first object is here very early in the oscillatory cycle, shown here. But now we do the same analysis for the second object. And now we see, again, more information during the polarizing phase of the oscillation. But now it's shifted to a later phase of the depolarizing depolarizing cycle, part of the cycle. So essentially, this is what we found is evidence to support this idea that the brain keeps ensembles separate from one another by virtue of this juggling act. The brain, 30 times a second, is going object 1, object 2, over and over again, 30 times a second. But more than that, you notice this is not some of you musicians in the room may notice this is not a straightforward juggling act. This is actually a syncopated rhythm. What the brain is doing 1, 2, pause, 1, 2, pause, 1, 2, pause. So it's 1, 2, 1, 2, like that. And what that means is that there's an explicit code here in the brain of the order of the two objects. The first object is always after the pause, and the second object is always after that. So this may provide a way of the brain to actually order thoughts. And shortly after we published this paper, a group published a paper showing that schizophrenics show a decrease in high beta, low gamma oscillations in their brains. So if this truly is what's ordering thought, then if you decrease those oscillations, you're going to disorder thought. Now, one thing you may say is well, maybe this is all just an epiphenomenon. The oscillations are the brain. It's like the car engine humming. The humming has nothing to do with the function of the engine. It's just a byproduct of the engine operating. So the way neurophysiologists have addressed this issue for years and for single neurons is you look at error trials. What happens when the monkey makes a mistake? What happens when the monkey makes a mistake is the phase effect goes away. Now, for this particular experiment, there was two types of errors the monkey could make. The monkey could either choose one of the wrong objects, or it could choose the correct two objects but do so in the wrong order. The latter, the order errors, were more common than the object errors. Unfortunately, we didn't have enough data here, enough error trials, because the monkeys are very good at this task. We didn't have enough error trials to separate out those two errors. But I suspect that the reason why these phase effects are collapsing is because on many of the trials, when the monkey went to the stimuli in the wrong order, the phase effect reversed. So we're now redoing these experiments with more electrodes to get enough error trials so we can parse out these two types of errors and see if the phase effect essentially goes with the monkey's behavior. If the monkey thinks 1 is first and 2 is second, the brain goes 1, 2. If the monkey makes a mistake, it goes to 2 first and 1 second. And the brain should switch over to this 2, 1, 2, 1 mode. That's what we hope to see. So from this, we learned that the dynamic formation of neural networks by synchrony may underlie cognitive flexibility. And multiple thoughts are simultaneously held in mind by oscillating them in a phase of one another. That's the suggestion of this. The brain does this juggling act. And this juggling act is two things. One thing, it keeps the ensembles from colliding together, so it keeps them separate, so they don't actually inadvertently bind and you get a jumble of thoughts, but it also can help order thoughts. But the other thing that I find interesting about this phase coding effect is that it may explain one of the biggest mysteries of conscious thought. Why is it you have a limited capacity for conscious thought? I mean, you can hold a lifetime of experience in your head. But for some reason, you only can express thoughts, at best, average person could hold 4 things in mind simultaneously. So why do you have this huge capacity to store information, but such a small capacity to express information, at least consciously? Why is that? Why can't you hold 1,000 things in mind? You have a lot of neurons in your brain, or 100 things in mind, or even 20 things. Why is it only about 4 in your average person? Well, maybe because if the brain is using, the brain needs conscious thoughts are flexible. Cognition is flexible. If the brain needs a flexible format to form ensembles to help form conscious thought, it may have to do so by virtue of these oscillatory dynamics, which I'm arguing binds networks together. Neurons that hum together wire together as long as they're humming. It's a dynamic format for forming networks. If that's the case, the downside of it is you've got to pack all the information you need into one single oscillatory cycle. Or actually, because all the big money happens on the polarizing phase of oscillation, you actually got to pack all the information into half an oscillatory cycle. So any communications, if the brain is using oscillations as this communication network to control which networks form, at least for high-level cognition and flexibility, well, the downside of that, it helps with the flexibility, but the downside is very limited capacity for expressing thoughts. And that's it. Thank you for your attention. I'd like to thank the people in my lab who do all the hard work. This work was Evan Anzelatis' work on the category stuff. Tim Bushman, my genius postdoc, now a professor at Princeton. And Eric DeNovellis did the ensemble work with Tim. And Marcus Siegel and Melissa Warden did the phase coding work. And what we're doing right now, I should have mentioned the phase coding work, is we just completed an experiment where we're loading the monkey's working memory up to the monkey exceeds capacity. And the prediction is that we'll explain the monkey's limited capacity by seeing this phase coding effect break down. And if we see a relationship between the monkey's limited capacity that's explained by phase coding, I'll submit to you that we'll have found the first neural correlate of consciousness. Because what's the most obvious and objective thing you can say about conscious thought? there's limited capacity. So if you can explain why there's limited capacity, you will have found the neural core of consciousness. I won't be able to explain whether you feel love or see red the same way I do, but I can tell you how a thought becomes conscious, hopefully. So thank you very much.
8 minutes for questions. You all
right? Those ensembles before, did they change from block of trials to another block of trials for color and orientation? Was it the same ensemble, or was it different ensemble for color among blocks of trials?
It was the same ensemble. I mean, we wouldn't have seen
100%.
Yeah, well, we wouldn't have seen these effects if there wasn't some consistency across blocks of trials.
Well, it would be 90% consistent.
Oh, I didn't do that calculation, but they seem pretty robust. Every time we went back to the same trial, we saw the same pattern. And we do these effects by averaging across trials and across blocks. That's how you pull out these effects. So if there wasn't some robustness to the ensembles, we would never have pulled out these effects to begin with. But I don't have an exact answer for how robust they are.
With random, you would be able, but I mean, there is more question. Thank you.
Sure.
So if like synchronous oscillations are what are driving like the formation of the ensembles, is it possible for a neuron that's, say, not connected at all anatomically to a network to be part of an ensemble so long as it's tuned to that frequency?
That's a great question. One possibility is that some of the communication in the brain takes place if you truly believe the oscillations play a role in brain function. One possibility that some communication may take place in the brain just by virtue of electromagnetic conduction. You have all these dendrites and axons all crunched together. There's a lot of opportunity there for just pure like oscillations here affecting a neuron nearby, even though they're not physically connected. So that's a possibility. Not a lot of evidence for it, but it's a real possibility. I think it's a real possibility. Some of the neurobiologists who made this opinion. Yeah.
I was wondering about the synchronous oscillations being a mechanism for separating ensembles, right? This is kind of what. But if that's it seems circular though. That doesn't. What tells those neurons to oscillate at that point? You know what I'm saying?
Well, if I had the answer to that, I'd have the Nobel Prize. I would figured out the brain. So there's a bunch of answers to that question. One is that, well, we think the prefrontal cortex essentially what its function is of the brain is to build models of a task, essentially. Build models of the goal-relevant structure of the world so you know how to behave in a lecture or how to graduate from Dartmouth. This is what the prefrontal cortex does. It builds models of goal-directed behavior, and somehow it's using that to direct traffic in the rest of the brain. How it gets the right pattern of oscillations to the rest of the brain, that's a great question. One possible hint of that comes from Sabine Kastner's recent work, who showed that in the thalamus there's lower frequency theta oscillations that act sort of as a carrier wave for the cortex. So all the higher frequency stuff in the cortex synchronizes together. They ride on top of these lower frequency synchronizations in the thalamus. So maybe the lower frequency oscillations for the thalamus, which has this nice topographic connections to the cortex, it may provide a carrier signal that tells which parts of the brain to the cortex to synchronize together. Now, if I knew exactly how that worked, wow. Yeah, sure.
So there's one, you've been looking at separate ensembles among the same set, right?
Yeah.
And so two separate thoughts. But I was wondering what's the relationship between this and say like metaphorical thought or like, insight, right? So you have like one ensemble buzzing and then you lead to something new that's similar to it.
Yeah.
Is that, it seems like this could be a good mechanism.
Could be. I think of that as a problem of generalization. If you can generalize, like you figure out what's common among all cats or all dogs. Well, that's just a way of throwing away details and leaving behind the essence, which, by the way, maybe categorization is a way which the brain may deal with limited capacity, because if you have a limited capacity for conscious thought, you want to throw away details and just leave the essence. You want to make use of whatever information space you have. So I think of things like that, analogies, metaphors, where you put two things together and get a new thing. I think of that as more this process of generalization, where you carve a new ensemble out of overlaps of existing ensembles. But of course, that's just hand-wavy speculation. You can tell by the way I'm waving my hands.
Do you ever see desynchronization phenomena? And is there a way for desynchronization to be functional? You think that that could happen.
Well, I mean, I guess this phase coding effect isn't strictly desynchronization because essentially they're still synchronized. They're just offset from one another. We saw this decrease in synchronization in the orientation network after this blast of alpha. So after the orientation network oscillated in alpha, all of a sudden there was a decrease in synchrony there. A complete decoupling, no, there's always some baseline level of synchrony in these neurons, just a matter of level of synchrony. But how can you disrupt synchrony? Sure, there's ways of doing that. I mean, there's ways of putting, nice thing about rhythms is that they can be reinforcing. So you have two networks resonate together, and they'll tend to stay together. Even if the frequency bounces around a little, they'll tend to change because they self-reinforce one another. But then with just a little bit of manipulation, you can cause them to decouple, cause them to desynchronize. How exactly that happens, what's directing it, again, I don't know. That's it's a high-level question that I don't know if we'll ever get to in my lifetime. Yep.
So you see decoupling of single unit with the local theta. Do you similarly see during error trials, you see decoupling of single units with theta? Do you see like breakdown of overall theta during these error trials? Like if a component of the engine breaks down, does the engine keep humming, or is there a misfire?
You do. So like in the ensemble experiment, the first experiment I told you about, I was talking about some new stuff here, so I wasn't sure how long the talk would go, so I didn't put it in. Turns out I could have had time for it. But what we find is that there was a correlation between the strength of the alpha and the strength of the beta and the monkey's reaction time. If the alpha was stronger, the beta tended to be stronger in the other network, and the monkeys reacted quicker. If on a given trial the alpha was weaker, the activation of the other network, beta, was weaker, and the monkey's reaction time was slower. So we saw this nice correlation between not only between the strength of alpha and beta, but the latency, the onset of alpha and beta, and the monkey's reaction time. Which again, when you see these kind of correlations between the phenomenon and behavior, that's the kind of thing that our neurophysiologists have used for single neurons to justify the role of single neurons in behavior. So it's no different here. You see all sorts of correlation with errors, correlation with reaction time, et cetera, and so forth.
So you mentioned this distinction between the anatomical connections sort of being like the roads and the oscillatory synchrony being like the traffic cop. I was wondering, was there any sort of like systematic time or activity in which the anatomical connections are more, like the neurons that are anatomically connected are going to be particularly synchronous versus ones that are not necessarily? So like I'm talking about sort of like the space across, say, prefrontal cortex,
Sure.
in which neurons are oscillating, the sort of dispersion.
Yeah, that's a difficult, if not impossible, question to ask right now, because I would have to know what the anatomical connections are of all these neurons in the living brain, which can't do. I'd like to think so, but if I could answer a question that's sort of related to that, is that I think there's also a relationship between the oscillations and the building of anatomy, building of the road and highway system. Because why do we keep seeing beta, high beta, low gamma over and over again? Well, it turns out high beta and low gamma are good frequencies for putting spikes into that window for spike time independent plasticity. Okay, so the phase differences you tend to see in high beta, low gamma, just put the spikes within that little window which fosters LTP, which enhances LTP. Lower frequencies or desynchronization, especially lower frequencies, may take them out of that, purposely take them out of that window. And so it prevents memories from forming. And an experiment we just finished, I didn't get a chance to talk about, which maybe I could have, is that we recorded from the prefrontal cortex and hippocampus while the monkeys are learning new things. And what happens when the monkey gets the trial correct, there's an increase in beta synchronization between the prefrontal cortex and hippocampus. When the monkey gets the trial incorrect, makes a wrong guess, there's an increase in lower frequency theta oscillations. So if that maps on to LTP and LTD, the animal gets the trial correct, turn on the gamma or high beta, low gamma to foster LTP. If the animal gets the trial incorrect, put in low frequencies to get them out of that spike time. So at the very least, there won't be any new memories formed. It may even foster LTD. There's less evidence for that. But at least it gets the spikes out of that window so the monkey can accidentally form a memory of a wrong thing.
Time for one more question?
Yeah.
So what do you think of different frequencies within gamma band and gamma band? So like high beta and low gamma bands. So
Oh, yeah.
that correlates with distance between those neurons. So
Yeah, great question. So my colleague Nancy Capella at BU has been very interested in different bands of gamma. And well, one thing is that I think you just hit on part of the answer is that one thing that may determine what frequency synchrony you get is how close the neurons are together. Local circuits oscillate faster because there's a shorter duty cycle because they're physically closer. Longer circuits, macro circuits, tend to synchronize at lower frequencies because the duty cycle is that much, the neurons are more separated. And that's maybe why we tend to see lower frequency stuff in the subcortex synchronizing the higher frequency stuff in the cortex because the distances are larger between the subcortex and cortex. So that may be part of it. But I think we have to be very careful not to equate frequency with function. Beta is not always going to be the same thing in every brain area. Theta is not always going to be the same thing in every brain area. It's like saying neural activity is the same thing in every brain area. They probably have different functions in different brain areas. So beta and theta may play a role in forming networks in some areas. They may form a role in plasticity in other areas. What determines the exact frequency, who knows? I mean, Nancy Capel thinks that low gamma is particularly important, and it depends for cognitive stuff, and it depends on a balance between excitatory and inhibitory neurons. And you always get that same frequency range because it depends on this architecture of excitatory and inhibitory neurons hooked up in these recurrent loops. So that's why you always get that around the same frequency range for all these tasks, which makes the phase coding effect important because if you can't separate them in frequency, you got to separate them in phase. But one last thing I'll say about it is that there's work from Matt Wilson's lab at MIT who had the rats running around two different mazes. Rats running around maze A, running around maze B. And then he records from the CA1 layer of the hippocampus, and he listens at, I forget the exact frequencies, but listens at a higher frequency, and he could tell what location the rat was in the maze. If he listens at a lower frequency, you could tell which maze the animal's in. So that's an example of how you can use this frequency-based coding, frequency-based communication to communicate two different messages at the same time from the same neurons, just by virtue of which frequency you're listening at. So I think there's a lot of richness here for exploitation in rhythms for exploitation by the brain.
All right, there'll be a reception now outside of 202. Thank you.